Happy afternoon, everyone. I cordially welcome you all to our GarbhaGudi Institute of Reproductive Health and Research, GGIRHR. Myself, Darwin, Business Development. It's my honor to take over the platform for today's CME webinar. We proudly welcome all the speakers, doctors, and students who gathered virtually. General instructions: mobile phones on silent. Please turn off your cameras. For any queries, please drop it in the chat box or please interact during the interactive session. Let's begin today's CME with a short note on the journey of GarbhaGudi. GarbhaGudi was founded in 2011, where our progressive work has led to the progression and implementation of the latest medical technologies and methods to overcome male and female infertility. GarbhaGudi IVF Center is known for its extraordinary and immense success rates, ethical treatment, affordable cost with world-class infrastructure and human touch. Now, GarbhaGudi is spanning over a decade, who is still serving as a ray of hope for thousands of couples. Recently, GarbhaGudi IVF Center is awarded with Bangalore's fastest growing IVF center by Radio City and also ranked third for its best treatment in South India and eighth place across India. GarbhaGudi, which is a chain and new generation infertility treatment hospitals with state-of-the-art infrastructure and cutting-edge technologies like IVF, HICSI, PGD, etc. address this ever-increasing infertility issues. With the backing of such a capable and strong organization, GGIRHR is providing the foremost training to budding clinicians, embryologists, andrologists, and other staffs to mold them for the challenges of infertility treatment. GGIRHR is affiliated with Rajiv Gandhi University of Health Science for Fellowship in Reproductive Medicine, IMA-AMS for Fellowship in Infertility, GarbhaGudi University for Post-Graduation Diploma in Clinical Embryology. We also provide ultrasound scan course and other residential and non-residential fellowship with GGIRHR certified. Students at GGIRHR are trained with tips and tricks, procedure and protocols followed at GarbhaGudi IVF Center, which is ART guided, has been unbelievable success rates. Now we're glad to inform you all that we are spread with 7 centers across Bangalore in serving thousands of infertile couples. Without further ado, let's begin today's CME. Our first speaker for the day is Ms. Rakhi Raj, who is an embryologist at GGIRHR and GG IVF Center, Hanmanagar. She is well trained with her laboratory skills and guidance the number of students in the field of embryology. Today, Rakhi is here to talk on Embryo Culture System. Over to you, Rakhi ma'am.
Thank you, Darwin. Happy afternoon, everybody present here. So today I'll be sharing quite a little information regarding the embryo culture system or what is actually happening inside our IVF labs. So what is it we usually do behind the screens? So what happens to patients' gametes? So it is nothing but in a short way, if I can say, we are just going to mimic the nature in our IVF labs. So, coming to today's talk, the topics or the things that I'll be covering up. So, it will be what are we trying to mimic inside our labs? So, what are all the components of embryo culture system? So, what is this culture medium? What are all the culture conditions that we require? And the last would be a take home message. So, what is the in vitro embryo culture trying to achieve, or what are we trying to get or gain inside our IVF labs? So one thing is balanced nutrients. Second thing, optimum culture conditions. Third, prevention from external contaminants and oxidative stress. And then removal of metabolites from immediate vicinity and constant motion. Okay, so these are all the key points that we will be looking for in our culture system. So, what happens generally in a natural scenario? What are all the things that we require, or what are all the things, or the reactions, or maybe the components or the compositions that the gametes require in our female reproductive system? So, I will be talking about a pre compaction stage and a post compaction stage as well. So, as you can see here in the picture, So here I have shown you a picture wherein we can see the implantation, the stages of implantation, wherein from the ovaries we get the oocytes, and from the vaginal area we get the after, I mean post the intercourse, yes, we get the sperm. So the viable one, normal viable sperm is going to hunt the, I mean whatever, is going to find the oocyte in the fallopian tubes, and it is going to fertilize the ovum. Okay. So once the fertilization happens, there are like different stages for a gamete to become a baby or a fetus, a complete healthy baby. So what are all that stages? So first thing would be a pre-compaction stage, that is from day 0 to day 3. What is this day 0 and day 3? I'll be coming into that. And the next set would be the post-compaction stage, that is from day 4 to day 6. So in this pre-compaction stage, if I can say, so this pre-compaction stage is nothing but the embryo or the gamete when it fertilizes. So the day it fertilizes, it is called day 0. And then when it comes to day 3, that is the cleavage stage embryos, it is day 3. So it when it reaches to a cellular stage, or maybe a 8 cell stage, or a compaction stage, that is called day 3 embryo. So post compaction is once the embryo compacts, so all these cells will form a ball, a mass of balls of cells, and it becomes a, I mean a ball of cells. So from that to a differentiation of the cell, that is the blastocyst. So obviously the blastocyst has two kinds of cells, that is the ICM and the trophectoderm. So till that, it is called day 6. So during this pre-compaction stage, what are all the components that the embryo requires in order to become a healthy embryo? So these are all the requirements that the gamete needs. So one thing is a low biosynthetic activity, low oxygen consumption, pyruvate preferred nutrients, non-essential amino acids only. So here during the pre-compaction stage, it depends on the maternal genome, and it forms individual cells. So those are called as blastomeres. Okay, and it has high sensitivity, and the temperature requirement is around 35 degree to 36 degrees Celsius, and pH is around 7.94. So this is regarding the pre-compaction stage, or if I can say the cleavage stage. Next, coming to the post-compaction stage, high biosynthetic activity, high oxygen consumption, glucose is actually preferred. Both here in the pre-compaction stage, it is only non-essential amino acids, but as the embryo grows further, as it expands, or the number of cells is going to increase, so it also requires more amount of amino acids or more amount of nutrients. So both essential and non-essential amino acids is required, depends on the embryonic genome. Okay, and transporting epithelium, two distinct cell types, that is the inner cell mass and trophectoderm forms, as I said, the blastocyst. So the temperature here is around 37.2 degrees Celsius. This is approximate temperature, and the pH varies around 7.2 to 7.3. So these are all the requirements that the gamete requires to form a healthy baby. Next, the culture system. So what are the components that we require, or the conditions that we require for a embryo culture system? The first thing, the main thing, is the embryo culture media. Second is the embryo culture dish or the embryo culture vessel. Next is the type and the quantity of oil that we use. Next is the gas phase, the main thing. Temperature and humidity. Incubator, the type of incubators that we use. The air quality, and of course the embryologist and the gametes. So coming to the embryo culture media. So what are we going to see here? The first thing, what do we expect from a culture media? What, I mean, if I can say, if we are culturing our gametes in these particular media, what does the media do? What it has to actually do to the gametes? That is our expectations. And the history of the culture media, how, from where did we start, from where did this culture media idea actually started? So that also we are going to see over here. And what are the key components in culture media? First thing, the expectations from culture media. First thing is to ensure a proper embryo growth. Second thing, decrease embryonic stress. third thing, to maintain a gamete viability. So these are the three main important things that the components that we expect that the media gives to the gametes. So the history of culture media. So it's all started in 1929, where the Lewis and Gregory, they actually cultured rabbit embryos in plasma. Next, from there, it developed into a mouse embryo, which was cultured in blood clot. And it next was the 1949, where the Hamnot. So he actually found out Krebs Ringer's bicarbonate solution. So he cultured the mouse embryos till blastocyst stage in this particular media. Next is the Steptoe et al. So he found out Ham's F10 media. So this was a little bit advanced than the Krebs Ringer's bicarbonate media, wherein he actually cultured human embryos till the blastocyst stage using the Ham's F10 with human or fetal calf serum. Next the advancement was done in 1978, which was actually again by Steptoe and Edward, the team who actually developed or introduced the IVF treatment into the world. Earl's simple salt solution. So they developed this particular media where they added the pyruvates, so which the embryo actually required in the pre-compaction stage. So they actually advanced the media by adding pyruvate supplements into the media. So this was when they actually became successful in producing the first IVF baby. So the next one is 1984, that is Menizo. Adding serum albumin ensures non-adherence to the glass surface. Next, again from 1990 onwards. So again, it was from the Steptoe and Edward team. So they actually made the media even more simplified and optimized media to bring in this co-culture idea. Okay, so there from there itself we have got from them, or maybe from Steptoe and Edwards advancements, or what do I say, if I can say the invention itself, from them itself we have got two types of media, that is the sequential media and the single step media. So I'll be coming into these two different types of media. So what are all the compositions of a embryo culture media? So what does this embryos actually require inside this media? So these are all the components that we require. The first thing would be the water. So that is a distilled water. Next is the basic salts, carbohydrates, amino acids. Again, amino acids of different kinds. That is the essential amino acids and non-essential amino acids. Fatty acids, EDTA. So EDTA will act as a chelator, and vitamins and antioxidants, proteins, antibiotics, of course, yes, so that it prevents contamination, and growth factors. So these are all the key components that has to be present, or that will be present in our culture media. So the same thing will be present in different concentrations and different stages inside our female reproductive system as well. So the new generation media, that is our advanced or the modern media, will have even more, or the even more added components in it. So media composition, as I've already mentioned, so these are all the it is the same thing. So again, the composition is the same. Next is the types of media for embryo culture. As I already told, it is a simple salt. So it comes from the history. As I've already spoken there, it is started from a simple salt solution, goes to a complex solution. Then we have a one-step culture and sequential media. Okay, so these are all the types of media that we had. Next is the differentiation, or maybe the comparison, if I can say, between all these commercially available or the modern medias what we have, the ready-made medias. So what are all these salts that are actually present in those kind of medias that I have put here? So it is just the comparison between the brands. So one media system that from the GeneMed is not, GeneMed is nothing but a brand. So one media is nothing but a one-step culture. Sequential, you can see that G1 and G2. So G1 is nothing but the pre-compaction media, and G2 is post-compaction media. So what are all the components that we require in that particular media? that is given here. So now I'll be coming to the culture media types. What are all the types of culture media? As already said, it is one step and sequential. And how do we do that? How do we follow? Obviously, there has to be a set protocol to follow that particular media. So that is why we have two different medias. So what are those? So I'll be coming into that. First thing would be a sequential culture. So what is this sequential culture? So the name itself suggests two types of cultures, or maybe 2 days of cultures, if I can say. So it was developed to accommodate the dynamics of embryos, nutrition, and to mirror the environment of female reproductive tract. So here, what are we trying to do? We are trying to mimic the female reproductive tract. So in a female reproductive tract, I can say the fallopian tubes, the uterine walls. So each stages from the fallopian tube till the implantation stage, it will be having a various concentration of different components. So in order to mimic that, we have something called as sequential culture media. The embryos are exposed to gradient nutrients as it passes along the fallopian tubes. The culture media is subsequently changed to allow for optimal conditions for the extended culture to the blastocyst stage. These media are formulated to address the changing requirement of the developing embryos, thereby reducing the cellular stress and limiting the need for the embryos to undergo adaptation to grow. So, few embryologists or maybe few doctors also prefer this kind of media. So, what are we trying to do here when we are doing everything out of the body, that is in vitro? So, a few of the embryologists will also prefer mimicking the same idea as the fallopian tubes, or maybe same idea as the female reproductive system. So that is why this sequential culture system was introduced. So the basic principle which we can say about the sequential culture media is, it is called back to nature. Okay, so back to nature is nothing but we are going to mimic the fallopian tubes or the uterine cavity components. So it will, the same gradient medias and the same components will be provided by our sequential culture system to the embryos, so that they grow it in a very viable manner. So these sequential culture medias will, the cleavage stage embryos use pyruvate. So here the energy source is pyruvate and lactate and non-essential amino acids for protein metabolism. Further, the stages. So the post-compaction stage will have glucose as a main source and essential amino acids for protein metabolism. So there are two different kinds of media, of course. So that is why in a pre-compaction stage, we have different components, and post-compaction stage, we will have different components. So that is why it is called back to nature principle. Next is the single or a monostep culture media. So this was used to support zygote development till blastocyst stage. It allows the embryos to choose what it wants during pre-implantation development, provides continuous support to the embryo throughout extended culture, hence no need to refresh the media, has optimized energy substrates like D-glucose and L-lactate, supporting in uninterrupted continuous culture. It is defined or designed to reduce ammonium buildup through the use of stable glutamine source. It has high magnesium calcium ions ratio to limit detrimental stress induced calcium influx into the embryos. So that is why it is called the let the embryos choose. So the main principle or the main motto of single step culture or a one step culture is let the embryos choose. So this one step is like, we are not going to change the media according to the stages of the embryos. It is only that it is one droplet of a media of that particular micro droplet, and the embryos until day 6 it will be cultured in the same media and same droplet. So here in this media, the components, it will be like all the components will be provided in this media, so that the embryos will itself choose this in a pre-compaction stage. If it needs pyruvate, or if it needs glucose or lactate or whatever it is going to take up itself. Next, post compaction stage, it requires glucose for what do I say, the energy source. It takes up that only, and it will give up the extra product outside. So the embryo culture protocols. As I already said, the sequential media and the single step media. The sequential media, one thing is there are different brands and different kinds of medias. So one thing is fertilization media, cleavage media, and blastocyst media. So in fertilization media, we'll be culturing the post ICSI oocytes, and on day one it will be again fertilization media, so that the zygote will form. From fertilization media, you will be changing it to on day one. So you will be changing it into a cleavage media, which will have all the necessary requirements which are required for a pre-compaction stage. Then on day 5 or a day 4, you will be changing it to a blastocyst media, which will have post-compaction stage components. But in case of single step culture, no need to change any media or no need to refresh the media. One single media will be having all types of components which is required for a healthy embryo growth. That's all. So sequential media is actually an interrupted culture, but a one step culture is a uninterrupted culture. So there is no fear of or if some embryologists, if they fear of pH change or temperature fluctuations or maybe contaminations, so they will obviously prefer for a one step culture, wherein they actually culture it till day 6 without disturbing the culture. That's all. So next would be the embryo culture strategies. One type is single embryo culture, and the second one is group culture. So what is this single embryo culture and group culture? First thing is single embryo culture. So individual evaluation of embryo morphology and rate of development, depletion of necessary substrates by other embryo if cultured in a group. This method ensures that the environment surrounding each embryo remains constant, reducing the risk of resource depletion due to neighboring embryos. So most of the embryologists will prefer single embryo culture, so that individual or individual embryo monitoring will happen, and there will be no fear of that one bad embryo will influence the other good embryo as well. So that fear also will be taken out. So next is the group culture. Group culture, on the other hand, capitalizes on the potential positive impact of embryonic interactions. Embryos cultured together can exchange embryotrophic factors, potentially enhancing their development. Group culture can also be logistically efficient, requiring fewer resources and incubator space compared to individual cultures. So this again, maybe few embryologists will prefer group cultures, wherein they are going to reduce the what do I say? Reduce the consumables also, or reduce the medias also. And few of the embryologists also prefer group culture, because their cumulative or their results of a good embryo will be that kind of percentage. It will be of a higher note. So that is why some prefer group culture itself. So the what are all the culture conditions that we require? So first main thing is the temperature. So the optimum temperature is 37 degrees Celsius. Exposure of embryos and oocytes to lower temperature is detrimental to their microtubules, thus risking aneuploidy in resulting embryos. Not even low temperature, not less than 37 degrees Celsius, not more than 37 degrees Celsius. High temperature, that is 39 degrees Celsius, can also disrupt the microtubule organization. Exposure to suboptimal temperature also affects transmembrane transport and metabolism and slows down embryo development. So that is why we will maintain a constant 37 degrees Celsius temperature from the start, that is from the ovum pickup till the embryo transfer. So constant 37 degrees Celsius will be maintained. Next, coming to the gas phase. So gas phase is nothing but what are all kind of the gases that we use inside our IVF lab. One thing is CO2, which we require is 5 to 7%. Again, the CO2 might change according to the altitudes. Okay. So the incubator types are chosen depending on what kind of atmosphere will be used for the embryo culture. That is atmospheric or reduced oxygen. Incubators installed with gas mixing system, that is a trigas incubator, where a single gas connection, CO2 inlet and a CO2 sensor to enable regulation of gas input. If the altitude is not taken into account, the incubation atmosphere with a setting value of 5% O2 or even lower can cause anoxic conditions for embryos. So always when we set the percentage, the altitude has to be taken care of. As the altitude rises, the CO2 percentage also rises. Within each incubator, embryos are grown in a microenvironment that includes a combination of temperature, that is 37 degrees Celsius, humidity 90%, and a mixture of three gases: carbon dioxide, oxygen, and nitrogen. Another very important factor is the growth media that is used to culture the embryos. So altogether, these are all the key factors or key components that we require for a healthy embryo development. Next is the ambient air quality, the most important thing in our IVF labs. So embryos are very sensitive to environmental toxicants, that is mostly the VOCs. VOCs are nothing but volatile organic compounds. So what are these volatile organic compounds? So these volatile organic compounds are just like your alcohols, and if can say another substance, maybe, okay, fine. So the main part is alcohol. So it can be produced because of our deodorants, or maybe the body lotions if we put, or the nail polishes if we put. So that is why we avoid all these factors while coming inside our IVF labs. Careful attention should be dedicated to general air supply, and purification measures should be taken to effectively remove VOCs. One natural thing that can be used for a VOC, to take off VOC, is activated charcoal. I have personally seen embryology labs wherein they keep activated charcoal powders so that it just absorbs the VOC particles which are there inside the IVF lab. So next thing is the buffering system. So the regulation. So this buffering system is a key point in a culture media because it actually regulates the pH. So it helps in maintaining a stable pH for a media. So the regulation pH is controlled by buffer, and so we can classify media according to type of used buffer. So again, according to the buffering system, the media is classified into bicarbonate medium, phosphate buffered medium, HEPIS buffered medium, MOX buffered medium. So these are all the different types of medium that we use. So HEPIS buffered medium and bicarbonate buffered medium. So HEPIS buffered doesn't require CO2 environment because it is already bicarbonate a media. So it doesn't require CO2 for a pH balance. Oil overlay also reduces the speed of CO2 loss and associated increase in pH. Next is the bicarbonate buffered medium. So the effect of CO2 has a different effect on the pH of the medium. So here the bicarbonate buffered medium means it requires CO2 to maintain a stable pH. So the CO2 concentration has to be like 5 to 7% or 6 to 7%. So what is actually this bicarbonate buffer or a buffering system? So there is a reaction that is going to take place inside our incubators. What is this reaction? So when we are actually in a humidified incubator, if we can say, we are going to add water inside the incubators. So that is actually going to react with the carbon dioxide inside our media, and it will react with the media to form a carbonic acid, which will dissociate to form a bicarbonate ion. So this bicarbonate ion will help in maintaining the pH of the media. So that is why the whatever medias or the bicarbonate medias like the culture media. So these culture medias are a bicarbonate media that requires CO2 for a pH balance. So the bicarbonate medias will be kept in a loose cap. So whatever tubes or the dishes that we keep with bicarbonate media, it will be closed with a loose cap so that there is carbon dioxide exchange within the media. So coming to a open and a closed culture system, one thing is a closed culture system consists of micro droplets, whether it can be 25 microliters or 30 microliters or even micro droplets of that, which is a lesser volume, which is then covered with liquid paraffin oil so as to maintain the osmolality, pH and temperature of the media. Next is the open system consists of culture media in a sterile multiple plates without oil overlay. So these two kinds of media will be actually made based on their particular principles. So open system requires a larger volume, that is 0.5 to 2 ml per dish. Next, closed system requires very minimal amount, that is the micro droplets, For closed system, we have it is actually covered with oil, so it doesn't require more amount of media also for a stable pH. This thing, I mean, for a stable pH balance. But for an open system, if we add more amount of media, the pH also will be maintained, and there will be less fear of the media getting dried up as well. So this is how we will be preparing a culture dish, and it will be overlaid with oil. So that is a liquid paraffin oil, and we have to pour the oil until the droplets are immersed completely. So this makes sure that there is a medium between the droplets and the embryos, so that it maintains a stable environment for the embryos to grow. So coming to my last slide, that is a take-home message. So if in doubt, stay towards lower temperature, that is our optimal temperature. Often simple things work best. Simplify a workflow. That is why we use a one-step culture media. So reduce gamete handling, touch less. So even in a sequential culture media, we are going to handle the dishes very often. But in a one-step culture media, it is only one media throughout. So the handling also is very less. If your cumulative results are good, then your culture condition must be good. Do not be afraid of change, but change only one thing at a time. So this was regarding my topics. So if there are any queries, you can shoot. If not, I will have a small quiz time, so that I will know better if you guys would have got my point or not. A question from Shalini Das. So doesn't the oil interfere with the growth of the embryos? So oil will never interfere with the growth of the embryos, because oil is actually kind of a protective layer to the embryos, because it is going to prevent the embryos or the media. So if there are any fluctuations in the media, so like pH variations or temperature variations, or if there is any contamination going to happen, so the embryos will never grow, correct? So the oil will act like a protective layer or a protective medium, wherein it will prevent the pH fluctuations, prevent the temperature changes, prevent the contamination, and it will also prevent this media dehydration as well. So oil is never going to hamper the growth of the embryos. So which system is preferred for culturing embryos? Closed system or open system? Always the closed system. We actually prefer closed system because it has a one protective layer, that, as I already told, the oil medium. But in open culture system, one main drawback is that yes, we are giving the embryos the amount of culture media that they require, in a higher amount itself. But the what do I say, the if I can say, the open culture system, the drawback is it there are chances of the embryo getting dried up as well, or there are chances of pH fluctuations or maybe contamination. So of course the closed system is always preferred. So any other queries?
Thank you, Raki ma'am. Your passion for the topic really came throughout your words, and you answered all your questions. Thank you for that, ma'am. Our second speaker is Dr. Shivranjini, who is a senior embryologist at GarbhaGudi IVF Center, Kalyanagar branch. She is a passionate and well-trained embryologist. She is one of the best trainee in the field. So over to you, ma'am, Dr. Shivranjini.
A very good afternoon to one and all present here. Myself, Dr. Shivranjani, and today my topic of discussion is about early embryonic development. Am I audible to everyone?
Yes, ma'am.
So, our human embryogenesis is a complicated process in which a fertilized single zygote, that is a fertilized egg, develops into a multicellular embryo. So, the fertilized egg, from the fertilized egg stage till 9 weeks, it is called as an embryo. So after that, it is called as a fetus till the birth. So what is a fertilized egg? It is nothing but the process of fusion of male and female gametes in the ambulatory region of the fallopian tube. So the fusion of male and female gamete produces the fertilized egg, so which is called the zygote. So the female and male gamete is 23 chromosome, containing 23 chromosome. It is a haploid cells. So both fuse together to form a diploid cell, that is a 46 chromosome, and then that single cell will be developing into a multicellular embryo. So that is all about embryogenesis. So now what is early embryonic development? Early embryonic development is the development of embryo from fertilization, that is from a single zygote till blastocyst. That is the first week of embryo development from fertilization to blast. So zygote is the beginning of life. So it undergoes several rounds of mitosis, that is cell division, and then as a result forms around 100 cells of blastocyst. So that is all about embryonic development, that is early embryonic development. So here, as I told before, there is several series of cell division. So it is also called as cleavage. So that means from one cell in blastocyst, there are 100 cells, but there is no change in the overall embryo volume. So as you see in the picture, the zona pellucida, which is outer covering of the embryo, all the 100 cells will be there inside the zona only. So it is all contained inside the zona pellucida. That's why the division is called cleavage. That's why we call it as cleavage embryos. From the fertilization to the blastocyst stage. After that, the embryo will hatch out from the zona, and then it will become bigger in size, and then the baby is born after 9 months of pregnancy. So in vivo, the sperm enters through cervical region, cervix, and then uterus, and then the oocytes are present in the fallopian tube, so that the transportation of male gamete is very important to reach the oocyte site. And in IUI, we expose the sperms on the uterus region. So from there, the sperm transports towards the oocyte sites. So two are very important for the fertilization. That is capacitation to reach the oocyte, and acrosome reaction is an enzymatic reaction where the sperm enters the oocyte, enters inside the oocyte. So the main important point for fertilization is the sperm-oocyte interaction, as it is shown in the picture. So this is how it happens. So sperm first penetrates the granule, the cumula cells and all, and then it releases the acrosome content, and then it invades inside the zona pellucida, and then the sperm contents enters into the oocyte. So three structures are important in entering the oocyte. They are, first of all, the sperm nucleus and the cytoplasmic content and the sperm centriole. So which is important for the development of embryo. So from the ampulla region where the fertilization happens, then the growing embryo will be transported from the fallopian tube towards the uterine part where the embryo will get implanted. This happens in vivo. Whereas what happens in vitro? Vitro means we are culturing the embryos in a petri dish. So in embryology lab, we culture the embryos in both conventional IVF technique, also in ICSI technique. So there we culture the embryos from the retrieval of oocyte, that is day 0, till the blastocyst formation that occurs on day 5 to day 6 or day 6. So in conventional IVF and ICSI, there is only one difference. That is, there is no sperm oocyte interaction in ICSI procedure. Whereas in ICSI, we take one sperm and directly we put it inside one oocyte. So there is no interaction between the sperm and the oocyte. So what happens immediately after the sperm enters the oocyte? So the first and the foremost thing happens is the oocyte activation. So that is all because of the sperm content that is released inside the oocyte. So that factor is called the soluble sperm factor, that is a PLC zeta. So that will increase the calcium content in the oocyte, which is the calcium is released from the smooth endoplasmic reticulum. So that helps in the oocyte activation. That is the first and the foremost for fertilization to happen. That will lead to resumption of second meiotic arrest. We know that oocytes are undergoing 2 arrests. So the first arrest is resumed, and then only we get a matured oocyte. So in the matured oocyte, if the sperm enters, the second meiotic arrest will be released. So that is all because of the activation of the oocyte. So that activation should be proper. It is a periodic and transient oscillation. The release should be periodic. So that will be in turn helpful in the chromosomal segregation. So that will lead to the euploidy, the normal 46 chromosomal content of the embryo. So the next step happens is a sperm nuclear decondensation. As I told before, the sperm enters, the main thing enters is a sperm nucleus. So the first thing happens after the nucleus enters the oocyte is a decondensation, and then only the pronuclear formation. So there are 2 pronucleus, one from the female, one from the male. And the 2PN formation is the first sign of fertilization. If at all there is no PN, there is no fertilization. So thus, that oocyte, which was having 23 chromosomes, now has 46 chromosomes. So became a diploid. So that is the beginning of that one particular embryo. So the next comes first single cell, diploid cell will be becoming a 100 cellular structure called the blastocyst. So the first division happens at around 26 hours post sperm entry, that is post ICSI. So for this, the main role is played by sperm centriole. So as the sperm enters the oocyte, the sperm containing two centrioles, one centriole will be entering the sperm, entering the oocyte, and that one centriole has to double, duplicate, and that determines a cleavage plane of that cell division, first cell division. So here, as I told before, the cell division is mitosis, not meiosis. So mitosis is nothing but a mother cell having a diploid chromosome, that is 46 chromosome, becoming two equally diploid daughter cells. So everything is same. Chromosomal content is same, that is 46, and cytoplasmic content is also same in the two daughter cells like that of mother cell. So this is how the mitotic cell division happens. So here I have mentioned karyokinesis and cytokinesis. Karyokinesis is nothing but chromosomal segregation, equal segregation between two cells. And cytokinesis is one cell dividing into two. The next is one cell dividing into two cell, and then the second cell division is 2 cell to 4 cell embryo. So that happens 44 hours post sperm entry, and then simultaneously the cell divisions happens. And then the next is third cell division that happens 68, that is day 3 post fertilization, day 3 post sperm entry. So as I told before, this cell divisions is called cleavage because there is no change. As you see in the picture, all the cells, 4 cells, 8 cells, whatever it is, they are contained inside the zona pellucida. There is no improvement in the volume increase in the overall embryo. So that's why it is called cleavage. So as I told before, the centriole will be determining the cleavage plane. So in that case, if at all the first cleavage plane is not correct, it's not in the center, so there will be demarcation in that cell blastomere blastomere size. So that indirectly will affect the overall furthermore development of the embryo. So that's the reason for uneven blastomeres and fragmentations. If one cell is dividing into two, in that case, if at all a fragmentation is expelled, that is cytoplasmic content wastage. So that also affects the embryo quality. So overall, the cell division also determines the embryo quality from the cleavage to blastocyst. On day 3, 8-cell stage, a very important process happens. So that is nothing but embryonic genome activation. So what is embryonic genome activation? It is nothing but, we have told before, the oocyte is injected with sperm and then it is fertilized and then it is becoming a blastocyst. So on day 3, till day 3, the maternal oocyte is supplying maternal mRNA that in turn will produce protein for the cleavage. So what happens after that? How long the oocyte will be supplying energy to the embryo? So in that case, there comes embryonic genome activation. There the embryo has to work on itself and it has to produce its own independent mRNA and protein for the further development. So that all depends on the genome of the sperm as well as the oocyte. So if at all the embryonic genome activation doesn't happen, then the further growth is arrested. So embryonic genome activation happen means that will further develop into a blast. So that is very important in case of our early embryonic development. So day 3 is for embryonic genome activation. The next is a 4th cell division, that is morula stage. This happens on day 4. So here in the first picture, you can see more than 8 cells. So morula is nothing but 16 to 32 cell stage, and it looks like a raspberry. So here 16 to 32 cells are adhere to each other. There is occurrence of compaction. There is presence of tight junction. So there is no demarcation between two cells, and then it becomes like a one cell structure, cell structure. And then the main important thing happening in morula is cell differentiation. That means the conversion of totipotent cells to pluripotent stem cells. So then what is a totipotent cell and what is a pluripotent cell? So a totipotent cell will give rise to both fetal cell and placental cell, whereas a pluripotent cell can only give rise to fetal cells. For example, I have mentioned this day 3 embryo, 8 cells are totipotent. That means if you take all the 8 blastomeres and culture separately, we can get 8 new embryos actually. So that one blastomere, which is totipotent, can give rise to one embryo, whole embryo, which will become a blastocyst again. Whereas if you take the pluripotent cell, it will never give a new embryo. It will only produce a fetal cells. So that is the difference between totipotent and pluripotent cells. And in morula stage, as I told before, differentiation happens. So the pluripotent cells are accumulated and patterned towards one corner, forming the ICM. So that's why in the structure of blastocyst, we say ICM is the one which is forming a fetus and forming a baby in the future. So the next trophoectoderms will be forming the placental cells, which will be supplying nutrition to the baby, the fetus. So on day 5, the end structure appears, that is the blastocyst. It is a fluid-filled embryo. And this bottom picture, you can see the structure of the blastocyst. The easily discernible prominent mass is the inner cell mass, which later on produces a fetus and the baby, which is pluripotent. And the trophoectoderm becomes the placenta. And the fluid cavity is called the blastocele. And we can see the zona pellucida, which is present since from the oocyte stage. So why the blastocyst is filled with fluid? Why the blastocele cavity? So it all helps in supplying nutrition to the ICM and trophoectoderm, and also to help the embryo to hatch out from the zona. The embryo cannot stay inside the zona pellucida and then die off. It has to come out from the zona, and then only it can go and implant inside the endometrium. So the main function of the fluid-filled blastocyst is to hatch out. So here you can see, because of the sodium potassium ATPase pump, the water is pumped inside, water influx happens, and the blastocele cavity is formed, and it is filled more and more, and there is no water outflux because of the tight junctions, which is termed as desmosomes. There is always only water influx, not outflux. So at one point, if at all it is the water content is increasing and increasing, the water pressure increases, and at one point the zona pellucida, which is nothing but a non-living glycoprotein layer, a jelly-like layer, burst opens, and then the embryo escapes out through that opening. So that's the importance of blastocele. So now this is a picture of a hatched blastocyst. So now the hatched blastocyst will come out, and then by this time we would have transferred the embryo to the uterus, and then the oocyte, the blastocyst will be opposing that endometrial lining to have a nice site for implantation, and then the implantation happens. So hatching is very important for the beautiful blastocyst to come out and implant on the endometrium. So this how the early embryonic development happens. So as I told, that is a fertilized oocyte, zygote, and then cleavage happens, the mitotic division, and then blastocyst, that is on day 5 or day 6. Then implantation happens. I have told before that day 1, day 0 is a oocyte pickup. The oocyte inject sperm entry. So day 1, it becomes a day 2 cell stage, and day 2 it becomes 4 cells. So this is a normal mitotic cell division timing. So it takes around one day for the completion of the mitotic cell division. So mitosis just happens 1 to 2 hours, but the whole process of mitosis takes 1 whole day. That's why it takes from day 0 to day 5 for the development of blastocyst. And then timing, the rate of development of the embryo is also very important to determine the euploidy status of the embryo. Too fast growing and too slow growing embryos have the chances of being an aneuploid embryo. Aneuploid is nothing but the chromosome content is not 46. It can be one less or one more, one set of 23 chromosomes are not there. So it is not normal 46. Then it is called aneuploid. So we have certain time limitations for the development of embryos also. So usually we check here in the embryology lab the embryos on day 1 from the ICSI time, that is 17 plus or minus 1 for the presence of 2pn. That is the sign of fertilization. Then what is syngamy? Syngamy is nothing but the disappearance of 2pn. So in the 2pn, one is male and one is female. So one pn is containing 23 chromosomes. So another one is containing 23 chromosomes. They will now disappear, and then the 46 chromosomes will be together, and then the mitosis happens. The 46 will be like the chromosomes will become sister chromatids, that is DNA replication happens, and then it is going to divide. So that stage is called syngamy. That will happen 23 plus or minus 1 post ICSI. So early cleavage egg, that is the 2 cell stage, that happens 26 plus or minus 1 post ICSI. So day 2 is 44 hours from ICSI. Day 3 embryos, that is 8 cell stage is at around 68 hours post ICSI, and day 4 morula is 92 hours post ICSI, and day 5 blastocyst development, normally it is around 116 hours post ICSI. So this indirectly determines the euploidy status of the embryos, but the confirmation is by the biopsy. How the embryo is getting energy to cleave from day 0 to day 5 in our early embryo development. So first till day 3, the embryo consumes pyruvate for the energy consumption, for the energy to cleave. So after day 3, it is glucose which is consumed by the embryo for the development. So why pyruvate and why glucose in this 5 days? There is a demarcation. So this all depends of embryonic genomic activation. So as I told before, from day 0 to day 3, the maternal mRNA will be producing the proteins and that will be used for the cleavage. So in that case, the maternal cytoplasmic contents had mitochondria. So it will be taking use of the mitochondria. So the pyruvate will be going inside the mitochondria, and then the Krebs cycle, that is our electron transport chain, our tricarboxylic acid cycle, our electron transport chain happens, and then the the 24 ATPs, that is the energy source for that embryo. So because of this, the embryo is cleaving, having that energy to cleave. So till day 3, it is a pyruvate, which is the energy substrate for that embryo. After day 3, the embryonic genome activation happens, and then the glycolysis step is 10 steps. So the enzymes and all will be produced by the embryo by itself. So in that case, it doesn't have to get pyruvate from outside. It can produce its own pyruvate by metabolizing the glucose. So that's how the shift from pyruvate to glucose happens. As you can see, the glucose end metabolism is pyruvate. So in that case, we don't have to supply pyruvate separately from day 3. So that's how the embryo gets energy. So from glucose, it metabolizes to pyruvate, and that pyruvate will be helpful in getting the energy through Krebs cycle. So through glycolysis, the embryo gets 12 ATP as energy, and through Krebs cycle, it gets 24 ATPs as energy, and that's how the embryos are very nicely cleaving after day 3. So that's why there is a demarcation between the embryo culture system, as the previous presenter told. So that's how pre-conception, it is pyruvate. Post-compaction, it is glucose. So what happens in vivo? In vivo, till day 3, the cumulus cells, which is present outside the oocyte, will be supplying the pyruvate by metabolizing the glucose. So that's how till day 3, it is the pyruvate which is supplied by the cumulus cells. So after that, embryonic genome activation happens, and it directly gets the nutrition from the uterine and fallopian environment, and then the embryo will survive by its own. So for the early embryonic development, not only the nuclear maturation of the oocyte is important, the cytoplasmic maturation of the oocyte is also important. First of all, what is nuclear maturation? What is cytoplasmic maturation? So nuclear maturation is nothing but, first of all, the oocytes are arrested, as I told before, in GB state. So first meiotic arrest, and then maturation happens, and then only the mature oocyte have the capacity to get fertilized. Then what is cytoplasmic maturation? When it is immature, when it is GB state, all the cytoplasmic contents are accumulated in the center towards the nuclear area. And after it is matured, when it is a mature oocyte, the cytoplasmic content has to be redistributed throughout the oocyte. So that is also important for a normal embryo development. Overall, as we told before, an embryo is formed by the fusion of male and female gamete. So both are responsible for the development of the early embryo. So how the oocyte is affecting? The quality of oocyte, as I told before, the nuclear and cytoplasmic maturation affects the embryo quality and the development. So that will indirectly affect the oocyte activation process. Then resumption of second meiotic arrest will be affected. That will lead to chromosomal segregation abnormalities. And the main thing involved in oocyte is the maternal mRNA storage. So why? Because that is what is providing the components for the division till day 3. So if at all the storage is poor, then the embryo development will not happen normally. So moving on to the sperm quality, the main thing is DNA fragmentation. We can put any sperms inside it, but the sperm with a very good DNA content only will become a blastocyst. So that is all because of embryonic genomic activation. The embryo genome has both sperm compartment and oocyte compartment. So in that case, DNA fragmentation in the sperm will directly affect the development of blast and also lead to arrested embryo on day 3. And then the main important, the optimal culture condition. As I told before, in embryology lab, we culture the embryos till day 5 or day 6 till blastocyst stage. And it is very important to follow an optimal culture condition for that embryo to grow normally. So even if everything is fine, if the embryo culture condition is not good, we won't be getting a normal good quality embryo. So it all depends on the temperature, CO2 concentration, O2 concentration, and humidity and osmolarity and everything. For example, temperature fluctuation. The normal optimal temperature temperature is 37. If there is mismatch of, if there is any mild fluctuation, it will affect the mitotic spindle. As I told, there are multiple mitosis from day 1, from day 0 to day 5. In that case, multiple cells are dividing at a time. In that case, all will be having a mitotic spindle. That is nothing but the metaphase plate. From there only the chromosome will be cut into two. The sister chromatids will be cut into two, and that will be pulled to opposite pole, and then the two cells are formed. If at all the mitotic spindle, the microtubule is damaged, the pulling effect will not be there. So between the two cells, one will be having more chromosome and another cell will be having less chromosome. That will lead to aneuploidy, a cohort of aneuploidy. So that will in the end produce a aneuploid blastocyst. So optimal culture condition is also very important. So I wish to show the video about this early embryonic development. So here you can see the oocyte, and we have injected the sperm and appearance of 2PN. So one is from male, one is from female. So now this is called syngamy. That is disappearance of 2PN and then the early cleavage. This happens day 1, that is 24 hours post ICSI. So this is day 2, 4-cell stage, that is 44 hours post ICSI. Now it is 8-cell stage, that is day 3, 68 hours post ICSI. So now happens embryonic genome activation. So now the embryo will be independently developing into a blastocyst. Further development is followed by the embryo itself. So here the morula stage, you can see there is no cell-to-cell demarcation. And now here the differentiation happens. Here you can see on a 3 o'clock position that a huge mass is formed. That is the differentiation. So that will be forming the ICM. So the overall embryo structure is called the trophoectoderm. And the fluid-filled cavity is called blastocele. And you can see a clear zona pellucida. So that is a protective layer. So for the implantation, the embryo has to come out from the zona pellucida. So now we can see how it is coming out. So here you can see the fluid entry. The embryo is expanding. And ICM we can see clearly at 2 o'clock position. And then this is one other technique how the embryo hatches out. The fluid is filled and then the embryo constantly constricts, collapses and expands. So in that case, it is stressing that zona pellucida to break down. And then there is a weak point and then the zona pellucida breaks and the embryo escapes. And then that hatched embryo will be searching for the proper site of implantation, and then gets implanted if the soluble factor, everything is optimal. So that's how the early embryonic development happens from the zygote to blastocyst. So if there is any doubt, you can ask me. I can see one question. So what will happen to zona as the embryo grows? As I told before, the zona will be broken down and the embryo will be hatching out from the zona. So it is just a protective covering. As I told before, the one cell will be dividing. So in that case, if there is no zona, if there is no outer covering, the embryo will be dispersed. In the morula stage only, there is compactness. There is tightness between each of the cells. So before that, day 3, all the cells will be loosely attached. If there is no protective covering the zona, then the embryos will be escaping. So then there is no multicellular organism, fetus, nothing. So that is the importance of zona. And on day 5, the expanded embryo will be hatching out.
Ma'am, there are another 2 questions, ma'am.
Yeah, I'm seeing it. What's exact day of fetal genome formation? Genome formation is 2PN, that is day 1. That is 17 plus or minus 1 post ICSI. That means post sperm entry inside the oocyte. So that is 17 plus or minus 1 hour. So PGT can be done only in totipotent cells. See, we have 3 biopsy stages. We can do polar body biopsy. We can do blastomere biopsy. That is day 3 biopsy. Day 3 embryos are totipotent. And then next comes the day 5 biopsy. So in day 5 biopsy, we are doing only trophoectoderm biopsy. There is no such word called ICM biopsy. Because as I told before, the ICM will be forming the fetus. So if we go inside and take the ICM cells, then we are inculcating problem to the baby, future baby. So PGT can be done in a totipotent cell, that is day 3 also, and then in a trophoectoderm also. So there are 3 biopsy techniques. Gender stage, I'm not getting the question.
Ma'am, it is about gender explanation, I think, ma'am.
We have 2 gametes. Female gamete is a oocyte, and then sperm is a male gamete. They fuses together and then forms a zygote, which is a diploid cell, and then that will cleave and become a blastocyst. So the gender stage, it all depends on whether that particular sperm is having a X chromosome or Y chromosome. So if at all the sperm that enters the oocyte is having X chromosome, then it will become a female baby, female embryo. So if the chromosome Y is present in that sperm, which is entering the oocyte, so that will be having 44 autosomes and X and Y chromosome, that becomes a male baby. So it is already decided by the entry of sperm. So we can't change it. The gender cannot be changed. So it all depends on the chromosome that is carried by the sperm, which is entering the oocyte.
So much, Shivaranjan ma'am. Hope we answered all the
Thank you.
questions. And thank you, ma'am, for your all the questions we have answered. And thank you so much, ma'am.
Thank you.